The previous technical guide video provided the basic requirements and principles of potency. In this technical guide, some of the most important questions regarding potency are answered, and hopefully a number of misconceptions and misunderstandings explained. The importance and value of measuring potency cannot be underestimated. In 2007, HemoGenix was the first company to develop a potency assay, Halo 96 PQR, for hematopoietic stem cell products. This assay is now in its fourth generation. Other assays developed by HemoGenix continue to develop as more information is accrued. This and the following technical guides provide some of our experience and expertise in developing and using potency assays. So the first question to ask is, what is the intended effect or response? The intended effect or response of a traditional drug is often the dose-dependent clinical outcome. This may be ascertained during phase 1 or phase 2 clinical trials. For cell therapy products, there is a low probability that the intended effect correlates with a dose-dependent clinical outcome. In reality, this is in fact a rare observation, since the clinical outcome is usually the result of the intended effect. It is therefore necessary to differentiate between the intended response of the treatment and the clinical outcome of the treatment. For hematopoietic stem cell transplantation, for example, the intended response is the process of engraftment, but the clinical response is the time to engraftment, when neutrophils, platelets, and red blood cells are produced as a result of reconstitution. This can only occur once the active stem cells have been transplanted and engraftment has taken place. Following on from this is the second question: must potency correlate with clinical outcome? Potency must predict or correlate with the intended response, but this need not correlate with clinical outcome. In an article entitled Potency Assay Development for Clinical Therapy Products, an ISCT review of the requirements and experiences in the industry, it states that where feasible, attempt to correlate clinical outcome with one or more assays for biological function. If there is a clear correlation, it will be straightforward to select the final potency assay for pivotal studies and beyond. This is a low probability scenario. A direct correlation between measured function, potency, and clinical outcome is in reality rarely observed in early mid-stage development, and it is currently not a requirement. The FDA guidance for industry on potency tests for cellular and gene therapy products, published in January 2011, states that efficacy data from well-controlled clinical investigations can provide evidence that a product has biological activity and thus is potent. However, use of clinical study data may not be a practical method to quantitatively test for potency to release a lot. That is, clinical data may not be available prior to release of individual product lots. Clinical data may not be coupled to individual lots. The third question is, why must the active components be measured? The active constituents or components are responsible for the intended response, which should be predicted by the potency assay. If the cell therapy product consists of a cell mixture, regardless of the purity, then it is required to know which cells contribute to the intended response, and the specific biological function associated with those cells must be measured. According to FDA requirements, all active constituents should be measured. In practice, however, this is virtually impossible. For example, if stem cell potency is to be measured, technology is not available to measure every single stem cell population within a stem cell compartment of a biological system. It would also be financially prohibitive to do so. Studies at HemoGenix have shown that a minimum of 2 stem cell populations would suffice to provide the information required to release the product. Even this might be problematic for cell systems such as the mesenchymal stem cell system, which, although in many ways is similar to the hematopoietic system, lacks significant information to identify and measure more than one active cell population. What characteristics of the active components must be measured in a potency assay? In essence, virtually any characteristic that allows a biological function or activity to be measured that is specific for the active components responsible for the intended response can be used in a potency assay. For example, a characteristic of all stem cells is their ability and potential to proliferate. If stem cell populations can be detected and their difference in proliferation potential measured, this can form the basis of a potency assay. It is this property that has been used in Halo 96PQR and Luminesque 96PQR to measure the potency of hematopoietic stem cells and mesenchymal stem cells respectively. In contrast, a property measured as a result of the intended response would be retrospective and would not be a potency assay. Can a non-validated assay be used to measure potency? The answer to this question is no. Validating an assay means that quantitative information has been obtained and documented regarding the accuracy, sensitivity, specificity, precision, and robustness of the assay. To validate an assay, standards and controls must be available. If a potency assay cannot be validated, the results cannot be trusted. Since a potency assay is used, among other things, to ensure batch-to-batch consistency and provide criteria to release the product for use in a patient, it is necessary to produce the most trustworthy results possible. Can a potency assay be measured from a single sample point? No. To measure potency, a dose response of the active cell constituents must be performed and compared to that of the reference standard. The basic principles of measuring potency can also apply to cell therapy products, as described in the introductory video to this series. In that video, it was explained that for traditional drugs, the potency is measured by the horizontal displacement of samples to that of the reference standard, and that the linear portion of the dose response must all exhibit statistical parallelism to each other. Statistical parallelism is measured by the slope of the linear portion of the dose response curve. For cell therapy products, and stem cell products in particular, parallelism between samples will rarely occur. But as with traditional drugs, the slope of the linear regression of the cell dose response will provide the ability to measure potency by comparing the slope of the sample to that of the reference standard. Measurement of a single point at a specific cell dose to produce a quantitative response that has acceptance-rejection limits cannot measure potency, but is used to measure quality of the product. There is usually a direct correlation between potency and quality. Can the lack of sufficient sample prohibit a potency measurement? Lack of sufficient sample is not an argument for not performing a potency assay. The potency assay has several functions. It measures batch-to-batch manufacturing consistency and stability. It predicts product failure due to incorrect potency. It predicts whether a product can be released for use in a patient. And it predicts whether the product will produce the intended effect or response. The advantage of measuring potency to ensure the identity and biological activity of the active components prior to use far outweigh not performing a potency assay and relying on trial and error. When must a potency assay be established? Biological characterization of the active components that produce the intended response is essential to establishing a potency assay, because it is this information that will help develop the assay. Development of a viable potency assay should be initiated as soon as possible in the development of a new cell therapy product. Based on more detailed characterization of the active constituents or components that produce the intended effect or response, the original potency assay may change, or more than one potency assay may be required. If a cell therapy product has been developed without a potency assay, then it will be necessary to develop an assay based on the biological characteristics and properties of the active components that result in the intended response. And finally, why is a reference standard needed to measure potency? A potency assay cannot be established without a reference standard. The measure of potency is the potency ratio. The potency ratio can only be measured with a reference standard of the same material as the sample. And establishing a reference standard is probably the most challenging aspect of a potency assay. And this is now discussed in the third technical guide of this series. Thank you.